Hi everyone, and welcome to week 5 of Microbiology Lab. So this week we are going to be talking about the Kirby-Bauer antibiotic susceptibility test, and then this is also the week that you are introduced to your lab report. So to talk about the antibiotic susceptibility test, there are several different types of antibiotic susceptibility tests, and what they all do collectively is they test to see if a bacteria is susceptible to an antibiotic or not, which basically means can that bacteria be destroyed by that antibiotic? Is that antibiotic effective to treat that bacterial infection? So while there are many different types of antibiotic susceptibility tests, we're going to be focusing exclusively on the Kirby-Bauer test for week 5 and then also for your lab report. I'm sure you have some knowledge of antibiotics. We've all taken antibiotics at one point or another throughout our lives. Antibiotics are antimicrobial agents, so they are basically chemicals that can be used to destroy microorganisms such as bacteria. Antibiotics are actually produced by microorganisms. There are many different types of bacteria and fungi that produce chemicals that are known to kill off other bacteria. They're doing it as a defense mechanism so that those bacteria don't take their nutrients, but we've used to extract those chemicals from those bacteria and those fungi to use them medically to cure infections. For example, the penicillium mold is what makes penicillin. There's a species of bacteria within the Streptomyces genus that makes the antibiotic that we know as streptomycin. So they are naturally produced chemicals. I mean, we can chemically modify them to get them to work better, especially when bacteria start to show resistance to them in their natural form, but from the beginning, they are kind of just these natural antimicrobial agents. So like I said, we're focusing on the Kirby-Bauer test. This is a test that looks to see if antimicrobial drugs, which are antibiotics in this case, are effective against different microorganisms. So basically what you do is you put a sample of bacteria on a plate, and then you put these little antibiotic discs that we see here on the plate with the bacteria, and you incubate it for a period of time. And then once incubation is complete and you look at the plate, what you're looking at are these clear areas. These clear areas are a good thing. That's where this antibiotic was able to inhibit this bacteria from growing. So typically, if you see a nice big clear area, that's a good indication that that antibiotic is able to inhibit bacterial growth, and that would be an effective drug to use to treat an infection with that specific bacteria. Now, if we look at some of these, like this one down here, there's virtually no clear area. This bacteria, whatever it is literally growing right up on that antibiotic. That means that that antibiotic's not effective. Most likely, this species of bacteria is resistant to the effects of that antibiotic. So if someone was infected with this bacteria, you would never want to give them that antibiotic because they would not get better. So how does this Kirby-Bauer test work? Like I said, I already kind of give you a general description of it, but basically what you do is you take a sample of bacteria, whatever bacteria you're given, and with that sample of bacteria, you put it on a specific type of plate. So the Petri dish plates that we use for a Kirby-Bauer test are these Mueller-Hinton agar plates. It's just a specific type of plate that's used for this test. So when you put the bacteria on the Mueller-Hinton agar plate, you have to use a certain technique. So you don't use the street plate technique that we are familiar with. The technique that you do use, though, is called a bacterial lawn technique. So what you would do is you would take your sample of bacteria on a cotton swab, a sterile cotton swab, and you would go back and forth all over this entire plate. When you create a bacterial lawn, your goal is to get bacteria all over every square inch of that petri dish. You don't want any section to be left untouched by the bacteria. So by creating a bacterial lawn, you are really making sure that there is growth all over the entire plate of, or all over the entire Mueller-Hinton agar plate. The problem is, let's say you were swabbing bacteria on the plate, and you accidentally left this whole section up here blank. You just forgot to put bacteria there. And we put an antibiotic right in the middle there. When we look at it later on, we might see this big clear area and we might think, oh, that antibiotic works so good at killing that bacteria. But what we're not realizing is, oh, we just didn't actually put bacteria in that spot. So in a medical setting, you could end up giving someone an antibiotic thinking it's going to work when really the test was kind of false. It wouldn't work in that case. So it's very important when you're setting up this plate and you inoculate the bacteria onto the plate that you do use that bacterial lawn technique. So once the bacteria is already placed on the plate in that bacterial lawn technique, that's the point where you go ahead and you put the antibiotics on the plate. So this plate has a lot of antibiotics on it. For the Kirby-Bauer test that we're doing, you're only going to be responsible for 4 different antibiotics. But basically, you'll see this in the video that you watch of the procedure, you use tweezers and antibiotic dispensers to remove these little antibiotic discs from the antibiotic dispensers. They're really just like little paper filters that are infused with some type of antibiotic medication, whether it's penicillin or trimethoprim, whatever it be. So you basically put those on the plate. You do want to put them far enough away from each other, and you also want them far enough away from the edge because you don't want the medication to kind of mix in, as that could mess with your results. But you basically put the little disc on the plate, and then after that you incubate the plate for 24 to 48 hours before you can look at the results. And the results would be what we see down here. We definitely do see bacterial growth, but what we're kind of focusing on again are these clear areas. These clear areas do have a name. They're called the zones of inhibition. So the zones of inhibition are the clear areas that surround the antibiotic after incubation has taken place. And not always, but typically the bigger the zone of the inhibition, the more effective the drug. Some antibiotics do need bigger zones of inhibition than others to be effective, and we have a chart to kind of look at that. But typically, if you see a big zone of inhibition, that's a really good sign. So we would have like a big zone of inhibition here and here, but then there are definitely some that have little to no zone of inhibition, which is very bad. Little to no zone of inhibition typically means the bacteria are resistant to the antibiotic, so the antibiotic is not effective. A bigger zone of inhibition typically means that bacteria are susceptible to the antibiotic, which means the bacteria are able to be killed or growth is inhibited by the action of the antibiotic. So like I said, some antibiotics require a bigger zone of inhibition than others, so unfortunately you can't just look at the zone of inhibition and say, oh, it's pretty big, I'm sure it works. You actually do have to measure it and then look at a chart to interpret the result. So to measure it, it's so easy. You just take a ruler and you measure the zone of inhibition all the way across, and you will always measure it in millimeters. So you just measure it all the way across in millimeters, and then you look at a chart to interpret your results. So we will get to the chart in just one second, but before we get to that, just a little bit about that Mueller-Hinton agar plate again. You do need to remember that that is a specific type of plate that is used, as it'll come up in your lab report. This Mueller-Hinton agar plate is very standardized, so it's made professionally, and we do kind of want it to be made in a very specific way. It has to have a pH somewhere between 7.2 and 7.4, so generally you want this plate to have a neutral pH to be consistent with results. The plates have to be poured perfectly, so the actual kind of jello-like mix that's inside the plastic dish, you do want it to have a certain thickness. It needs to be 4 millimeters thick. If it's too thin or too thick, that does mess with the consistency of the results. So it's supposed to be 4 millimeters. The problem is if you pour the agar too thin, and let's say it's only 2 millimeters, what happens is the medication can diffuse too fast, and it can actually make the antibiotic look more effective than what it should naturally be. So you might give someone a medication and think it's going to work great, and then it doesn't actually work so well. If the agar is too thick, then the medication might not diffuse enough. And what would happen is the test would look less effective. So you might not give someone an antibiotic that could potentially work because of inconsistencies within your results. We also want to avoid excess moisture when we work with these plates. Kind of just think through the process. If there's excess moisture on the plates and you're putting medication in the plates, what happens when you kind of water down medication? It dilutes it out, so it might not look as effective. So these plates are standardized. There's a lot of things we have to watch for with these plates in order for them to get the appropriate test, because the Kirby-Bauer test is really important. It is a test that's going to tell us which antibiotic is going to be the best one to give a patient. So like I said, those zones of inhibition definitely need to be measured. You can't just eyeball it, but we measure the zones of inhibition in millimeters, and then we do look at that standardized chart. When we look at the standardized chart, we're looking to see if the bacteria are susceptible, resistant, or intermediate. Susceptible is a good thing. Susceptible means that the antibiotic is effective. The antibiotic is able to stop the bacteria from growing. That's what we want. Resistance is a bad thing. Resistance means that the antibiotic is ineffective. That means the antibiotic is not able to stop the bacteria from growing, and the bacteria continue to grow in the presence of the antibiotic. Then there's this kind of middle range intermediate category. An intermediate category means that the antibiotic would work, but it would typically have to be given at a higher dosage over a longer period of time. A doctor is never going to choose an intermediate drug if there are susceptible ones present, but if the patient has allergies towards the drugs that are considered more susceptible, or if they're on medication that would cross-react with the susceptible antibiotic, then a physician would settle for an intermediate drug. But it's never going to be the drug of choice, it's just kind of an emergency backup. So like I said, you measure the zones of inhibition in millimeters, and then you look at this standardized chart to see if the bacteria are susceptible, resistant, or intermediate to the antibiotic. Now this is an image from our textbook or our lab manual that we typically use, although you're not using it this semester. I still wanted to put it in here. So the antibiotics that we're going to be working with are chloramphenicol, Cipro, penicillin, and trimethoprim. Streptomycin and tetracycline are listed here, but you do not need to worry about those ones. But this has the name of the antibiotic. You don't need to worry about this section right here. The code, if you remember, those little antibiotic discs are really, really small, and we can't fit the entire antibiotic name on them, so they're all stamped with their little codes like C30 or CIP5. Disc potency. This is just how much medication is in that little disc. And then these are the ranges for the zones of inhibition. So it's so easy. Let's say we have trimethoprim, and we're working with like Staphylococcus aureus, and trimethoprim produces a zone of inhibition of 25 millimeters. So we measure it all the way across, it's 25 millimeters. All we have to do is see where 25 millimeters falls in this row for trimethoprim. Kind of ignore these little pencil marks. So anything 10 and under is resistant, 11 to 15 is intermediate, and anything greater than 16 is susceptible. So at 25, definitely susceptible. So Staphylococcus aureus would be susceptible to trimethoprim. It would be a good antibiotic to take. But let's say the zone of inhibition was 14. If the zone of inhibition was 14, we just see where 14 falls on here, right between 11 to 15. So that would mean that Staphylococcus aureus was intermediate to trimethoprim. That antibiotic could work if given at a longer dosage or a higher dosage over a longer period of time. And then let's say the zone of inhibition was 8 millimeters. Well, that's less than 10, so that means Staphylococcus aureus would be resistant to trimethoprim. So it's just finding the zone of inhibition and then seeing where that zone of inhibition falls for your specific antibiotic.